Many of you might be familiar with the plant-based Beyond Meat type products that have made their way into major fast food retailers such as Tim Hortons and A&W. But this isn't just a trend in the fast food industry. The global plant-based protein market is an industry worth $13 billion annually, and its growth is critical in meeting the growing food demand for an increasing global population. Many plant-based proteins come from crops we all know, including chickpeas. However, there are two major issues with chickpeas that really prevent it from reaching its full potential as a food source for human consumption. The first problem is that chickpea seeds individually don't actually contain very much protein. So you would have to grow a whole bunch of chickpea seeds just to get enough protein for one burger, which isn't very efficient. The second problem is that chickpeas can contain certain starches that make it difficult to digest. So if you've ever been a little gassy after eating too many chickpeas, if you know what I'm talking about, that's not actually your fault. It's because of a starch called raffinose that's present in the seeds. And in addition to making you gassy, the raffinose starch can actually make it more difficult for the human body to properly digest and absorb the nutrients in your food. The chickpea industry calls for a better line of chickpeas that is high in protein and low in raffinose starch, and that's where my job as a plant geneticist comes in. I can take a look at the chickpea genome and make the necessary edits to create an ideal crop line. In order to do this, we use the technology called CRISPR-Cas9. CRISPR-Cas9 is essentially a craft toolkit for DNA, so it allows us to make very specific cuts and edits to genes, allowing us to nullify or knock out these genes and render them non-functional. This allows us to manipulate the physical characteristics of the plant. So in order to address the first problem, the need for more protein in chickpea seeds, I took a look at the chickpea genome. I came up with a couple of genes that are thought to be involved in the seed protein accumulation pathway, and then I knocked them out using CRISPR-Cas9. And when we did this, the results were phenomenal. We were actually able to find that when one of the genes was knocked out, the seed protein content increased significantly. So that's problem number one, the need for more protein solved. The second problem, the raffinose starch that causes gas in chickpeas, is also another similar quick fix. I was able to identify the gene in chickpeas that's responsible for synthesizing raffinose, so as soon as we knocked that gene out using CRISPR-Cas9, no more raffinose, and that's problem number two solved as well. With all this research in place, the last thing we need to do is put it all together, and this can actually be done simply by breeding the high protein line of chickpeas that we've created with the low raffinose line of chickpeas. And once those two plants are bred together, this new line of chickpeas that we've generated is ready for growth in the field and ready for human consumption. As the plant protein industry continues to grow, my line of chickpeas helps us work towards a better and more sustainable future with higher yields and higher nutritional content. But on a more personal note, I'm really glad I'll just be able to eat as much hummus as I want without getting gassy afterwards.